Hi everyone, welcome back to my channel. It's Iris the Biology Tutor. Today I will introduce a useful analogy to help visualize the human gut microbiome and all of its dynamic interactions. As a reminder, the gut microbiota refers to the community of trillions of microorganisms, including bacteria, archaea, viruses, and fungi that reside in the gastrointestinal tract. These microbes are essential for digesting food, synthesizing vitamins, modulating the immune system, and protecting against pathogens. Check out my human gut microbiome video for more information. It may be useful to think of a healthy human gut microbiome like a healthy flower garden. Both require ample species abundance and diversity to thrive and are susceptible to invasive organisms. When gardens experience too much herbicide treatment, plant species can be wiped out. In humans, antibiotics act like herbicides to plants and contribute directly to decreasing bacterial species diversity, changing metabolic activity, and promoting antibiotic-resistant organisms. Broad-spectrum antibiotics target a wide range of bacteria. Typically, when a bacterial infection is suspected in patients, but the type of bacteria is unknown, or when infection with multiple types of bacteria is suspected, broad-spectrum antibiotics are prescribed. In fact, broad-spectrum antibiotics comprise more than 60% of the antibiotics prescribed by U.S. physicians. These antibiotics can kill or inhibit not only the pathogenic bacteria causing infection, but also beneficial bacteria in the gut. This nonspecific action leads to a reduction in the overall microbial diversity and a disruption of the balance between different bacterial populations, which can allow harmful bacteria to proliferate. When selective pressures are exerted on the gut bacteria, favoring the survival of resistant strains, certain bacteria can overgrow, leading to an imbalance and potential further infections and resistance. According to the CDC, United States healthcare providers prescribed 236 million antibiotics in 2022, which is about 7 prescriptions for every 10 outpatients, with nearly 30% of these prescriptions being unnecessary. The increased overuse of antibiotics has negative impacts on the gut microbiome and has other ramifications that I could discuss in a future video. Comment down below if the topic of antibiotic resistance sounds interesting to you. Eliminating plant species in a garden can allow room for non-native species to grow. In the garden, the plant invaders compete for the same nutrients which impede the growth and development of native species. Similarly, in the human gut microbiome, bacterial infections in the gut can occur when the healthy gut has become compromised. Clostridioides difficile, or C. diff, is part of the normal gut microbiota in a small percentage of healthy adults and infants, but can become pathogenic due to opportunistic gut disruption. When antibiotics disrupt the balance of the gut microbiota, reducing populations of beneficial bacteria that normally keep C. diff in check, C. diff can thrive, as it faces less competition for resources and fewer obstacles to its growth. As the bacteria proliferate, they produce potent toxins that damage the intestinal lining, leading to inflammation, fluid secretion, and cell death. This bacterium causes diarrhea and colitis, and is estimated to cause nearly 500,000 infections in the United States each year, with approximately 15,000 deaths annually. In the garden, invasive species can be eliminated through the introduction of wildflower seeds and soil. Wildflowers can compete for resources, enhance soil health, and disrupt the life cycles of invasive species, thereby contributing to native plant community restoration. An analogous case can be made for the method of fecal transplant as a treatment for severe gut infections. Typically, standard gut infections are first dealt with by discontinuing and citing antibiotics, if possible, then with targeted antibiotic therapy, and, if recurrent or severe, with fecal microbiota transplantation. A fecal microbiota transplant or stool transplant is a procedure where fecal matter from a healthy donor is transferred into the GI tract of a recipient. A stool transplant can initiate the restoration of a healthy gut microbiome by introducing a community of bacterial species back into the patient's gut. Similar to a healthy flower garden with plentiful and assorted blooms, a healthy gut microbiome, marked by both species abundance and diversity, supports critical aspects of host health. Abundant microbial populations enhance nutrient processing, immune activation, pathogen resistance, and gut integrity through effective resource utilization and competitive exclusion. Diversity provides a broad range of metabolic functions, immune system support, pathogen protection, and maintenance of gut barrier integrity, ensuring that the microbiome can adapt to changes and resist diseases. This balance fosters a resilient ecosystem that effectively processes nutrients, modulates immunity, and protects against harmful microbes, contributing to overall well-being. Thanks for watching this video. I hope it was clear and informative. Please like this video, comment down below on what else you'd like to see, and subscribe to my channel for more content.